Welcome to Fuzzybutts and Friends. Well, as much as it drives Fuzzybutts absolutely crazy all over this place, when they hear me say it, I am the host of this show, Yer Big Dog, Luke Robinson, back again with you this week. And look over there who we have back again with us this week. It's Ginger Morgan. She's our co-host, and she's also the executive director of the Puppy Up Foundation. Ginger, other than the weather, how are you today?
I am doing well, and I want to point out that November is Canine Cancer Awareness Month and Senior Pet Month. So, as usual, I always want to put a shout out to Lily because not only is she a cancer survivor, but she just turned 16. So.
Always get happy, sweet 16. I think I calculated that, and that's like 142 or something in human years. So she
Yes.
has certainly endured the test of time being a cancer survivor, and she's a fighter. And Ginger, I love that it's November. Well, I don't love that it's November and Pet Cancer Awareness Month, but it certainly dovetails into the conversation today, and Lily as well, because Ginger, I can't tell you how excited I am to have our guest on today and to be talking about this topic, because as you know it, I love science, I love technology, AI in particular. I'm a huge tech nerd because I believe it holds a promise of the future. And as we go into the Pet Cancer Awareness Month of the year, I envision a world where we have normal cancer rates, not these crazy super hyper cancer rates that we're having now, that we're experiencing now. As you know, Ginger, we think that there's 6 million new cases of cancer in dogs alone every year. That's just a frightening statistic. And that's in the US. That's not across the world. So we are witnessing a pandemic right before us. And I tell you, Ginger, as you know, I've lost 3 dogs to cancer, and so many times in the 15 years I've been doing this, I just wanted to press pause. I wanted to press pause and stop everything and stop the dying and the carnage until the technology caught up with us and we had solutions, treatments, therapies out there that could keep our companion animals alive. I'm going on. I'm very passionate about it. I apologize, but I just want to press pause. And I think our guest today might actually have a solution for that. So it's a tremendous honor, Ginger, to introduce Micah Mills with Cryopets. Welcome to Fuzzybutts and Friends, Kai.
Thank you very much. Happy to be here. And you'll have to let me know who Lily is. I'm not familiar.
Lily is my dog. She just turned 16. She's had 2 different types of stomach cancers and 1 mast cell tumor.
Yeah, 16. What
breed is she? She is beautiful. That's what we call her, the beautiful breed. She is a cross between, I believe, a chow for sure, but I believe golden retriever, chow and golden retriever.
I see. Yeah, so one of those kind of bigger dogs, right? And
yeah, she is. She's in our house where we have 3 Great Pyrenees or Pyrenees mixes, she's medium sized, but
right.
Her real healthy weight was about 53 pounds at the most, and she weighs about 45 now.
Yeah.
Well, we'd like to start. I'm sorry, go ahead.
No, I was just going to say I just have 1 cat myself, and she's about 12 pounds, so much.
That's exactly what I was just going to ask you, Kai, is that we'd like to start the show talking about living Fuzzybutts. Yes. And so you have a Fuzzybutt, you have a kitty cat.
Yeah. I have 1 cat. I've had her for about 11 and a half years now.
Wow.
So got her when she was just a kitten, from some farm in the middle of Utah. And funny enough, we named her Atlas when we got her, but at the time we thought she was a boy because that's what the person told us at the place, and she had long hair, long fur, so I just assumed that's what she was, right? And we went with it the longest time. And when we went to go get her spayed or neutered, as we thought, we got the paperback from the veterinarian that said "I'm a girl" written in all caps across the paper. And so from that point on, I don't know, the name, like, it felt kind of weird. I mean, Atlas I think could work either way, but I don't know, we kind of decided maybe we should come up with a new name, and I just always called her Cat because we didn't have a name for her, and that just kind of stuck. So she's just Cat the cat, and yeah, that's been my Fuzzybutt for the past.
Well,
Yeah,
I
years.
love the name Atlas for a girl. I think it's wonderful. I know that women can carry the weight of the world on their shoulders in our society, and so many do. And gosh, I wish they would shrug though, because especially when it comes to cancer. So that's so let's talk about your origin story, because I find this fascinating. I don't know anything about you. I do some due diligence prior to the shows, but I like to kind of keep an open terrain and just explore it. So Kai, tell us, how did you start Cryopets and get in this interesting business of cryopreservation?
Yeah, no, yeah, it's not a usual one to jump into. I grew up in a very very religious family, and a Mormon family. That's part of the reason that we're in Utah. It's kind of the one of the main places that Mormons like to gather. And moved here when I was about 12 years old or so. And I think it was around that time that I wanted to figure out what I believed, and I started sort of asking all these questions, and I ended up kind of pulling away from the Mormon church a little bit. But I think, like anyone, I really loved a lot of the things that I was promised from religion, right? Which was maybe dying, but not dying forever, at least, right? Having this sort of afterlife, and having being with the people that you care about forever, and this and that. I mean, it's obviously things that you would want to be real and true, right? So I was kind of in a weird spot where I was pulling away from religion, but I still, of course, wanted a lot of these things that it was promising, and honestly, life in a lot of ways felt just pointless without a lot of those things, right? And it was around this time that I was on YouTube, I think, and I came across this video from a Russian billionaire named Dmitry Itskov, and he wanted to make humanity immortal by 2045. That was his goal. And his sort of organization was called the 2045 Initiative. And he was more into sort of like mind uploading, life extension, kind of that kind of area. It's not really where most of my attention goes, but it was just that idea of maybe getting a lot of things that I was promised through religion, and accomplishing those through science. And that kind of I mean, that really sparked everything for me. That was my main interest. So
Well, I understand. I grew up also in a Christian, in a similarly religious background. And actually, my mom was deeply religious. My dad's a doctor, so a man of science. So I grew up part science and part religion. I love that I was experienced to both worlds. And it's always been sort of they haven't been mutually exclusive, but I've never figured out how to unite the two. And so I find that religion and the promise of eternal life is a lot of the inspiration behind this. But I got to tell you, I believe, I mean, I could never consider myself what most people consider a good Christian, or a good Christian, but I know that I felt faith until I lost my first dog, Malcolm, to cancer. And after that, I stared what Nietzsche called the abyss. I stared into the abyss, and I stared at that for so long. I've been down in the abyss for so long and found no meaning. So I feel like I kind of understand that feeling of emptiness, of void, of there is no promise, there is no afterlife, so therefore life has no meaning. And it's a tough place to be. And I'm glad that you, and like you, I've looked to science, I've looked to creativity and other things to find answers. So I think it's a really a powerful origin story. So a billionaire, a European billionaire, had developed the technology for people. So how long ago? When did Cryopets begin? When did you form the company, and where are you at now? Talk to us a little bit about about the company itself.
Yeah. So I started kind of thinking about Cryopets. I think I came up with the idea for it back in 2021. And I didn't pull the trigger on it until 2022. And this is, this is quite a few years after I found, Dmitry Itzkov and what he was working on, right? I mean, I grew up with no money in the middle of Utah. So, I always wanted to make an organization or a company that was working on something in this space. But, the barriers to entry there are, insanely high, right? So I just, back then I did what I was naturally good at, which was just computers. And I was, I love gaming. And so I kind of merged those two worlds. And, the first company I made back in 2012 or something was just a Minecraft server, right? And I worked my way up from there, and I've been, a serial entrepreneur for the past 10 years, sold a few companies in, the cloud computing space and built companies sort of all over tech. And then, yeah, it was around 2021, after my most, recent company that I was in that spot where I could take on something like this, right? I could get over those barriers now. I had the experience and the resources and everything necessary. So I was, I wanted to, map out the space, figure out, what needed to be worked on, what was, oh, there we go. Is that Lily?
That's Indiana.
No, this is Indiana.
I see. Yeah, that's a large
But
dog.
he's 12 and he's experiencing peripheral neuropathy. And so we see the horizon. Unfortunately, we see a horizon at the end. So I'm at that point where I'm scared. So I'm sorry to interrupt your train of thought. You were speaking about barriers to entry, and you're at that point. By the way, cheers to being a serial entrepreneur. That's part of my background. Don't know if I'll have time to get into that, but I admire you for doing that. That's I know exactly the grind, what kind of grind that is, and that you've done it and accomplished it more than once is quite an accomplishment. So you're right. You kind of have to be in a mental, financial, psychological, spiritual place to be able to take on a tremendous undertaking like this, Kai, which is the barriers. I just thought about it. They're never mind the regulatory and legal, just the psychological from a market standpoint. You got quite the challenge ahead of you.
Sure. Yeah. No, there's it seems like, I mean, most people aren't going to focus on world scale problems because they're worried about the rent next month, right? And it's so it's very tough. You almost have to solve every single one of your problems. And this is a lot of what I tried to do was like, I almost close myself off from a lot of things. I metaphorically, but also in a very real way, like, shut all the curtains and just tried to block out the entire world and solve every single one of my problems until I could get to the point where I solved most of my problems, right? And then I could focus my energy sort of outward. And that's when I sort of started this. But yeah, I wanted to map out the space. I was interested in the work that's going on in the aging field, right? I was still interested in, the mind uploading field, what people are doing there. Obviously, pretty far off. There's there's cool work going on all over the life extension space. And I ended up making this sort of, I saw this tweet from Balaji about sort of tech trees to map progress in different fields. And I just love that idea. And I ended up making a tech tree sort of, very high level, nothing, super detailed, but just sort of a tree mapping out what was going on in the entire life extension field. Right. So I saw people working on aging from a bunch of different angles. I saw people working on AI and making artificial brains. And I had this entire column on the left for preventing death. And the best way that I saw to do that was through pausing biology, right? And I visited, Alcor in 2016, who is, they're an organization that's been trying to kind of do stuff in the cryo field for about 50 years now. I visited them, and I guess I thought by the time 2021 or 2022 came around that things would be much, much further. But it's just such a small field, and when I mapped it out, I found that there was no funding, no startups in the field. It's just a few organizations that have been around for decades that are just running off donations. And it was shocking to me because it always struck me as the most important part of that entire tree. It was, it's not something that just directly targets a single cause of death or even a prominent cause of death. But if we can pause biology, we can target almost every single cause of death. And so I see it as sort of this technology that really directly targets the death problem, and yeah, that's really where I wanted to jump into that field. So.
Well, I'm a big fan of yours, and I'm a supporter of yours, and I want to help you out in any way I can, because I think that Ginger and I Foundation and the tremendous, wonderful community of pet parents who've lost their loved ones to cancer can give you feedback, be supportive, and help you along in any way that we can. And so I truly want to preface our conversation about that. But yes, but you know what's interesting is that everything starts. So you have approached this market research, actually trying to understand the problem of pausing biology, and realize that we really haven't. The tree doesn't go very far. So it's a good market to address, and it has tremendous future potential, which we'll get to in a second. And I know some of the players that were early into this. I was friendly with Sam Barshop, the Barshop family in San Antonio, and they started an aging institute, I'm sure as you know. And when I was a businessman in San Antonio, I was helping high tech and biotech companies back then. And so I know that we've made some research, but yes, it's really been dependent upon billionaires that through solipsism and just their own self-interest in wanting to be alive forever, that's driven the research, and it's a good thing. But I wanted to talk briefly about. I find it interesting. I want to get your thoughts about the billionaire who was invested in adrenochrome. He's that billionaire that wants to be young. He's using his son's blood. Are you familiar with that case?
Yeah, Brian Johnson. Or...
That's right. Yes, and so I followed his research as well, because again, even if it's driven by the fact that he just wants to stay alive and it's self-driven, everyone can benefit from the data that's derived from the science, right? So there is some science out there, but like you were saying, it's still so young and still so new. Sorry, go ahead, please.
Yeah, no, I'm definitely very familiar with Brian Johnson. We're actually, both from Utah. He's also an ex-Mormon, funny enough. So there's, he's a controversial figure without a doubt. I think you find that a lot in longevity.
Absolutely.
A lot of people are, not really comfortable with that idea yet. It's the longevity space. Part of the reason that I decided to go into cryo is because I think the progress in longevity is so unbelievably slow.
Right.
And after looking at it for so long and talking with so many experts, even the top, biogerontologists in the space will even say that, curing aging in our lifetimes is just a pipe dream, right? And that's not what you want to hear from the experts in the aging field, right? So it's an incredibly important problem to work on. We need to continue working on it. It should, get even much more funding than it has now, but there is a reason that I, decided to prioritize cryo. And, Brian Johnson, as far as, I love his, I love the don't die, banner that he has on everything. I love that sort of that way of thinking. And I think we do need to be, taking our bodies very, very seriously and doing all these tests and all of that. As far as how it's going to apply to everyone else, I mean, they are N of one experiments, right? And there's obviously a lot.
But we need N of ones, though. N of one is actually, that's how most of it starts out, as an N of one.
Yeah. So it's some of the examples that he's setting, I'm definitely on board with. And
I didn't mean to speak badly of him. My apologies if I sounded like that. I was just saying typically billionaires. I've known a few billionaires in my lifetime and multimillionaires, and I just most of that is driven through self-centered interest. And it's important to distinguish that even if that's the case, the N of one is still valuable. That data is still valuable. You're familiar with David Sinclair and his approach is that aging is a disease state. And so we need to approach it. So you're in this really fascinating end of the market, if you look at it that, or industry, if you look at it that way, of pausing biology. And so that's what's so cool about what you're doing is it's great what the Sinclairs of the world, that's great what they're doing. The Brian Johnsons of the world, that's great what they're doing. But I want to specifically focus on pausing biology. And I know that what you're working on is going to be critical to the future, because sometime we're going to be traveling far differences out in the universe, and we're going to need to figure out a way to pause biology. That's just a reality. So the seeds of what you're working on is absolutely essential to the future. So on that note, let's delve into the science, because I want to nerd out with you. So where are we at? Let's start off. I call it the science of cryogenics. Is that the correct term for it, Kai?
Yeah. I mean, when we're talking about, cryogenics, right? Where we're referring to that low temperature, right? And cryobiology is, it's a fairly wide field, right? I mean, we there's things that we cryopreserve all day long, all over the world, right? There's for reproductive reasons, for research, different types of cells, tissues, these are things that we cryopreserve all the time. So when we're talking about, specifically whole bodies or brains or in the way that we're trying to do it, that's when it's referred to as cryonics. But a lot of times we just say cryo, and refer to it like that. But
Okay, so give us a kind of a snapshot of where are we at in the science of cryonics and cryopreservation.
Yeah, it's a field that's come a long way, and it's not a field that has, really anyone that's doing a lot of science communication. So there's people who think we're just throwing people or animals in freezers and hoping for the best, right? When, the technology and the procedure is very, very complicated. But we've come a long way, especially in the early 2000s up to now. There's been a ton of really, really incredible breakthroughs. One of probably the most prominent one is some research that was done by Greg Fahey, who is he leads the 21st Century Medicine company, and he's developed the cryoprotectant that sort of the top cryonics facilities use, including us. And he was able to cryopreserve a rabbit kidney, bring it all the way down past the glass transition temperature, very, very low temperature, recover it, bring it back, rewarm it, and then retransplant it into a rabbit. And it was able to be, viable and work, and that rabbit was able to survive and live its life. So that's obviously a huge jump from, doing cells.
How recent? I didn't know about that study, and that's amazing. How recent was that?
That was in 2009. It was published.
And so it was transplanted in the rabbit, and the rabbit lived normally for X number of years that they followed it.
Yeah, so the rabbit, they kept it alive. I think it was like 43 days or something in that range. They ended up euthanizing it for, to conclude the study and to, do more tests on the kidney and see how it was, reacting. But yeah, they basically decided that, yeah, like this rabbit would survive indefinitely with this kidney. So.
Right. Well, we know that Kai, we know there's a biological precedence, though, for cryonics, though, because there are some biological species out there in nature that exhibit it, and they do it every year, as a matter of fact.
They do. Yeah.
Which ones are those?
Yeah. So there's probably the most, famous one for that is the tree frog. It's a species of tree frog. You can find it in Alaska and other areas. And yeah, I mean, it will, when winter comes around, its heart stops beating, its blood stops flowing, and it stays in that state through winter. And then when spring comes around, it thaws out and it comes back to life, right? Which is obviously incredible that you can see this in nature. It's pretty unbelievable. They have sort of, some natural cryoprotectants built into their to their biology that we do not have. So I wouldn't try it, I wouldn't go out and try to, but there's, we see this as well, even with a lot of fish. You can walk over a lake and you can see a fish, frozen in the close to the surface, and then it'll thaw out and a lot of times be just fine. But even in humans, there's been cases where, people have drowned in a lake, a frozen lake, and they're able to be recovered an hour later or even even longer than that. So it's pretty incredible. But yeah, those are some of the frogs and some other ones are.
So we know that there's a biological precedence that can take biological species down past. You use a term, I don't know if I got it right. Glass transition, is that what?
Yeah. So we can. So
what does that term mean?
So it's funny when articles come out about cryonics or cryopreservation. It's always X company is freezing pets or freezing people. And actually the vast majority of the work we do in cryonics is to prevent freezing.
Okay.
We do not want freezing, because freezing means ice crystal formation. And when
Okay.
ice crystals form, they crush cells from the outside.
Right.
They expose them to lethal amounts of salt and so on. So it's not pretty. It's not something you want. So you know, what we do in cryonics is we actually replace all of the blood in the body with cryoprotectant, which is sort of like a medical grade antifreeze.
Right. Right.
And then when you bring it past that temperature mark, it depends on the cryoprotectant. For M22, I believe it's like negative 123 degrees Celsius. Then it's what's called going past the glass transition temperature. And what that does is it sort of forms this glass-like state where ice crystals don't form. And that's what protects the structure, and that's what really leads to the theory that we can bring these.
Right, because I know enough about the biology to I know that the ice crystallization, when you're thawing, if that's the correct term, or bring it back to temperature, that it actually lyses the cells and it causes just massive damage and disruption. So that's that term, glass transition temperature. That's a facet. So you never want to reach that point. And these antifreeze fluids, are they akin to what the biological species have? Does their blood convert or form some type, some type of coolant? I guess is, for lack of better, antifreeze.
Yeah, sure, yeah.
I'm using non-medical scientific terms. I apologize, but
no. In a way, yeah.
Okay.
It's similar. You know, when we're talking about the temperatures that we go down to are obviously much colder than winter is going to give you, right?
Okay. But it's that coolant that protects.
Yes. So it's in a lot of ways, it's comparable in the sense that we can bring biology down. It's paused in a sense when it comes to these frogs, right? But when we talk about liquid nitrogen temperatures, I believe 1 second at our temperature that we're at is like 10 million or 110 million years at liquid nitrogen temperature, something like that. So it's we're talking about completely halting biology, right? Whereas just winter temperatures aren't going to your steak's only going to last so long in the freezer or in the fridge, right? So yeah, we have to go very, very, very cold. Which makes it difficult. There's a lot of things that come with that. There's different levels of thermal stress and damage that's done that we have to prevent at those temperatures.
Let's talk about there's so many things, so many questions I have. Every time you talk, I have more questions. But let's talk about the closest that we have to a success with this technology is it the kidney experiment that you're referring to, or is there any more recent?
So there's there's been organ work here and there, right? So a lot of stuff with organs. There was some really exciting research that was with a technology called persiflation. This was I think it might have been around 2014 or something in that range, and they were able to recover pig hearts. Using persiflation, and I think persiflation was sort of attempted back in the 90s to keep hearts viable for longer during transplant, but and I think it was mostly scrapped back then. But one of the biggest problems in cryopreservation of larger tissue structures is that cryoprotectants are toxic at room temperature, right? So there is a certain level of damage that's being done there as you're cooling the body down and as you're putting the cryoprotectant in. But it's a necessary evil, because we need the cryoprotectant to prevent ice formation, which obviously prevents a lot more damage, right? And they sort of theorized that with persiflation, which is basically perfusing the organ with helium, actually, they were able to cool the organ down much faster and use much less cryoprotectant. And that was able to almost circumvent that problem. And they had some really promising results in pig hearts. And company ended up going under. Like I said, there's really not a lot of funding in this type of research, but that was some exciting work. There's also been success in brains, which is probably the most important for what we're working on, right? In 2016, Robert McIntyre and Greg Fahey as well, which was the guy who did the rabbit kidney experiment. they were able to, practically perfectly cryopreserve the ultrastructure of a rabbit brain, using what they called aldehyde stabilized cryopreservation, which was a huge win as well, because it showed that not only can we cryopreserve kidneys or livers or hearts, but also the brain, which is obviously very, very important for what we're doing. I mean, if you get all your organs except for the brain cryopreserved well, then we're kind of missing the point of the end goal here. But and then
that is the most important. Let's stick with that for a second, because I want to touch on that, because that is the thing. That is really the only organ that we matter, because we know that nature gave us, let's just say, our hardware is finite. Our hardware is flawed. Maybe our software is flawed too, but definitely our hardware is flawed, and it's certainly finite. And so the only thing that really matters to any of us, and humans, of course, is our brain, our memory, our recollection, our thoughts, our creativity, and maybe all the untapped potential. But with our companion animals, though, it's our brain, but it's also our personality, right?
Yeah.
Because if it's not, we're in a pet cemetery, Stephen King sort of scenario, right? Where you just have
get that comment a lot.
What's that?
I get that comment a lot,
I try to avoid that, because I take what you're doing very seriously, and I want you to know that. And so I do try to avoid all the easy ones, but it is sort of the crux of the conversation. It is the crux of the issue. It's applied more to humans, because my understanding, which is limited, is that some of the cryo experiments with humans, or back in the day, like with Walt Disney, is they actually decapitated they kept the head, right? Because that was the most important thing. Whereas we're not going to be looking at that with cryopets, with our companion animals, but still we want that personality. So the question becomes, how do not with what certainty, we have no idea, but what we want to accomplish with cryopets is not only the revival process, not only restarting the biological process, but ensuring that the personality and the companion that you love is still intact, right? That's the crux of it.
Yeah, 100%. Because otherwise, I mean, maybe we would dive into cloning more or something, right? You can clone your pet and get a new one, and some people do that, and it helps. But at the end of the day, your original pet, their memories, their personality, who they were, that's a totally different being, right? And of course, that's lost. So,
I've heard cloning has not had any success. I'm sorry to interrupt, but I wanted to just put this in there because I wanted to ask you a question. I've heard cloning has not had really any success with replicating any personality traits, or is that true?
Yeah, no, it's completely true. Yeah, it's a totally different, animal. It shares physical traits.
Literally.
But it's like it's something that I think it's really interesting technology for things like having a clone of yourself that you can get a transplant from whenever you need it, or some other kind of more far off areas that I'm really interested in. But I wouldn't clone my cat, because it wouldn't be my cat.
Right. I wouldn't either. But I do have an interest actually in pausing the biological process. I sincerely have an interest in it. So Kai, where are you at? So we know we've had some success, some successful trials at the organ level. Never, correct me if I'm mistaken, never an intact species revived 100%, correct? We've never done that before. Is that correct?
So there's I think one of, and it's good for this topic that we're on as well, as far as keeping memory and personality and so on. There was an interesting I think it might have been in 2016 as well, maybe a little bit later, experiment that was done by Natasha Vita Moore and Greg Fahey, who has been carrying this field for a very long time. He's doing so much of it, where they basically trained C. elegans, which is it's like a very small worm. It's used very often in scientific research. They trained it to sort of avoid a certain chemical, and then we cryopreserved them, brought them all the way back, rewarmed them, right? And after being brought back from cryogenic temperatures, it had kept its memory. It still remembered to avoid that chemical. And there were even pregnant C. elegans that they did that ended up having their offspring after they were brought back from cryopreservation, which is just interesting as well. It's just cool that we were able to, pause biology even of pregnant animals, right? And bring them back. Now, of course, C. elegans are very, very small, and we can take learnings from that, but of course we need to be working on larger, animals and mammals specifically, which is what we're focusing on. But yeah, just an interesting experiment that shows that at least with smaller, animals, that memory does survive going down to those low temperatures and being paused, right? And I will say, just because you mentioned it, unfortunately, Walt Disney is not cryopreserved. He was that's kind of one of the a lot of people mention Walt Disney when it comes to cryo, but unfortunately he was not cryopreserved.
That's a myth. Darn it. I'm a research guy, I'm a data guy. I almost always, if I use something, I look it up, and I look it up until I'm convinced I've got a good source, which gets harder and harder to find. But thank you for clarifying that.
In cryo, it's a tough field to find a lot of truth. Like I said,
but and you're gonna find this more and more so, actually, and we found this with our foundation, is that you can't rely on the press to accurately portray the importance of what you're working on and to get your value proposition out there. They do a horrible job of it. They're gonna mess it up. They're gonna use wrong data, and that's becoming increasingly true. So that's why I want this platform to be an opportunity for us to get the right data to pet parents out there, because I'm excited about this. I did want to mention, though, I was more of an H.P. Lovecraft guy than Stephen King. So and Pet Sematary, I was more of the Reanimator. That was my show, but that's more obscure. Most people don't understand that show. All right, so let's see. We walk us through, so we have a basic understanding of the pausing process. Walk us through the revival process. How do you propose to revive?
Yeah. So along with when it comes to revival, we're talking about a lot of basic science here. Right. And this is part of the reason that a lot of research in this area hasn't been done at, cryonics facilities in the past. It's two reasons. It's apparent that the most important part of this is being able to, cryopreserve and retain that brain structure. Right. Once we're down at those temperatures, we have practically unlimited time, as long as we can keep them in that temperature state, to solve the problem of rewarming, right? So I think that's part of the reason a lot of research hasn't been done in that area in the past, and also because it's a difficult problem, right? It's easier to put them in than it is to take them out, right? So beyond just cryopets, I have a foundation called CryoDAO, actually, based in Switzerland, and this is where we do a lot of the sort of the research that is a little more towards the basic science area and translational science in cryopreservation, but we have a strong focus on large tissue structures, right? We'll do organs, we'll do experiments with whole bodies and things like that. And one of the first projects that we're getting off the ground there is sort of a novel rewarming technique for large organs, where we use ultrasound technology, actually, and we surround the organ with thousands of ultrasound transducers, and it's almost like a very smart microwave, if you will, right?
Wow.
And so that's one of the experiments. Is that going to be the one that lets us recover entire organs? I'm not sure. I think it's promising. And persiflation, like I said in the past, really, really interesting. There's a lot of these technologies that are on the horizon that just need money and funding behind them. But yeah, so that's one of the ways that we're working on it now.
So you've invented a giant, like, microwavable defroster, essentially.
Well, so at CryoDAO, we fund researchers around the world that are working on these. So
right.
I can't take credit for the very smart microwave, but
you use the correct term, transitional research, that you hope one day can be transitional, applied to additional species, to other species as well. What do you think? What are the big risks? What do you foresee as the possible reasons for failure? If a pet parent comes to you and says, let's do this, and we'll get to use cases and other things here in a minute, but just if I come to you and say, I've got this in terminal case. My boy has terminal cancer. We've exhausted all treatment avenues. We know what's going to happen. It's only a matter of time. Um, well, first off, walk us through that process as though I've come to you and then I'm discussing this case with you, because I know this is what pet parents want to hear. So walk me through when I come to you and I give you this potential use case. I've got my case of terminal cancer with my kid. Walk us through that.
Yeah. Yeah. So it's, one of the big reasons that I, wanted to start with pets, aside from obviously my cat that's running out of time herself, right? Is that, I think the science is very, very real. And the reason it's real is because we can get lab quality cryopreservation. With humans, it's a very difficult process, right? That person dies of whatever. Usually it's a sudden death scenario. We don't get euthanized, of course, at least not in most places. And then they need to be declared dead at a hospital, and then they need to be transferred to the cryogenics facility. And a lot of times, so much time has passed that we can't even perfuse the body. We can't even replace the blood with cryoprotectant. We just have to do a straight freeze of that person. And so these are all, massive problems when it comes to human cryonics, and it leads to quality that just isn't there, right? When it comes to animals, a lot of times their deaths are planned, right? They get to a point where, it's the humane thing to do to euthanize them when they're in, too much pain to keep going. And so that really, allows us to get that lab quality cryopreservation, because we can go directly from euthanasia to the cryopreservation procedure on the same table, right? Because we aren't just a cryonic facility, we're an animal hospital, right? We're a hybrid of both.
Okay.
And part of that, reasoning, there's a lot of reasons for it, but one of the important ones is, in this field in the past, historically, there's been a lack of real doctors doing these procedures. And I've been very adamant about the fact that it should be only doctors that are doing these procedures, right? If this field believes that we can bring these animals back, then they need to be treated as still being alive, in a sense. They need to be treated with, the respect of having a well-trained real doctor, because the procedure isn't always straightforward. Sometimes, if the animal has some sort of ailment, if the animal has certain blood clots, these can complicate the procedure, and we need someone that's very well trained and knows how to still execute when that happens. So that's one of the reasons, we can get more into that as far as being part animal hospital. But I mean, I'm rambling. What was the original?
No, that's okay. These are
Yeah.
you can ramble, but you're always talking about questions, addressing questions or other questions
Sure.
I have. So I want to clarify that it's the point of euthanasia. That's where that's when I come to you, or obviously I come to you before we're ready to go, but know that there's no more quality of life and we're ready to go. And so from that point, then you take that over and you have the hospital facility and you have the veterinarians. Do you currently have that staff right there or set up, or is that something under works?
No, so we've been entirely in research for now.
Okay.
So it hasn't been a point to bringing on. I mean, we have DVMs that are assisting in constructing these procedures and so on, but
Absolutely.
we don't have surgeons or anyone around the clock yet, because the facility's not open yet. But
well, that wasn't even fair. I didn't even ask you that question. I should have started out. So where are you at because the company formation? I'm sure as you've started so many. That's critical. Where you are in that? Where are you in that sort of S curve of formation?
Yeah, so we're based in Utah. We're based in Salt Lake City, Utah. Part of the reason, obviously, I live here, and we know a lot of people, and I have talent here and things like that. But more importantly, Salt Lake City is the fourth safest city in the nation for declared by FEMA for natural disasters and things like that. So when you want somewhere that needs to last presumably a very long time and needs to be stable for a long time, Utah is just a fantastic place for that. So as far as building a cryonics facility, that was one of the main reasons that we wanted to be there. But
you've got some good research going on there too. I know that for sure. Yeah,
absolutely. Yeah. So yeah, that was one of the main reasons for Utah. But that's just going to be where the storage is. A lot of what we want to do is reach pet owners where they are. right? We don't want people to have to travel across the
Right.
country. And even, I mean, we blew up quite a bit in Spain, right? We have a huge community in Spain that's super interested in this, and
Wow.
you know, other areas around the world where it's obviously that would be very difficult, right? So you know, part of the reason that we're an animal hospital is it's sort of a proof of concept. You know, if I had to imagine where I want cryonics to be in the future, not only with pets but with humans. I mean, my parents are getting older. My grandparents are coming up on 90 now, right? You know, I'm incredibly lucky that they're all still alive, right? And as they get older, you know, you start to see they start having health problems here and there, and it's just I want to know that, you know, if I have a grandparent or one of my parents that ends up dying because of whatever reason, I would like to know that there's a hospital a mile or 2 miles or 3 miles down the road that can perform at least you know, the initial step of the cryopreservation procedure, which of course does not happen in hospitals today. And it's very difficult to get to.
Right. Well, so
You
so
know.
so yeah, I was going to say that because if you're looking to build a facility there, that's your limiting step of your whole operation, because pet parents won't even drive 2 or 3 hours to go see a board certified veterinary oncologist, even though Ginger and I tell them to every single episode. But so it would be, but so that's right. You understand how to scale. You've got to be there at you got to be there deployed decentrally, right?
Yeah.
So that's how to scale. So you have a location in Utah that's still in R&D. At what point are you ready to what's the inflection point, Kai, that you're ready to go and start open up the first satellite, let's call it a satellite, at the first pet hospital in Utah? How far away is that for you, do you foresee?
Yeah, so we, as far as, you know, we've figured out sort of all of the main engineering and, you know, science challenges that we had to get to this point, which was very difficult, and it took you know, it took quite a bit of time, because, you know, you can't Google how to cryopreserve a dog. Right? This is, you know, it's a lot of the work, you know, that's been done on humans, you know, wasn't crafted specifically for dogs or cats. There's up to 12 different medications that we use throughout the procedure, and there's different amounts of those medications that we have to use, because, you know, for humans, you know, you can get a general, you know, dosage, but as far as all these different species of dogs and cats and we have, I mean, we have hamsters, rabbits, horses, monkeys, you name it, on our wait list, right? So it's difficult, and we've had to craft this sort of in a way that works for all these different animals, obviously focusing on cats and dogs first. So we've gotten past most of the challenges there. There's a few more pieces that we have on the way that are just finishing up, and we should be able to perform the first procedures probably in the next 2 months.
Okay, so I guess I should have called that your beta site. When do you anticipate to have your first beta site? But you're going to do your first actual cryonics, your first use case. I guess this would be considered your first use case within a couple of months.
Yep.
And you'll do that there in Utah.
Yep, it'll be here in Utah. And
okay. Congratulations. I guess in order
you.
this, I'm assuming you're a bit nervous, but excited at the same time. What type of pet is it, if you don't mind me asking? That's okay.
Yeah, so I mean, so we have a lot of pets on our wait list. Of course, the first ones that we do all the way through from start to finish won't be ones that are off of our wait list. It won't be customers' pets, right? It'll likely be rabbits to start that are dedicated specifically for research, that are already, you know, we to do the testing,
sure,
on our pet owners' pets, right? We want to, you know, so that's what we'll be doing in the next 2 months. And then you know, once we're comfortable with the procedure all the way through, and we're in the actual you know, animal hospital, that's when we'll start doing it for, you know, those who are waiting on the wait list. So
got it. So I skipped over when you were talking about barriers to entry. We kind of skipped over regulatory and legal. So is your operation regulated by the USDA or FDA, since you're going through trials, or either or none?
No, none. So I mean, this is one of the things that was really promising to me about the field of cryopreservation is because, as far as the government is concerned, even with humans, these bodies they're dead, right? They don't have rights anymore, right? So so it puts us in a place where, you know, the research can move pretty quickly, right? There's, you know, if we're talking about aging drugs and trying to go that way, like these are things that take very, very, very long and take, you know, so many millions of dollars just to get through a lot of this regulation. Whereas in you know, in cryo, we don't have any of that. Now,
right, because you would,
you know,
almost like be like considered like the orphan drug exception, right? That it's a use case where there's an end of life or whatever. There's a need for urgency, right? That they streamline the R&D process for those reasons, right?
now if there was, you know, let's say there was a medication that we wanted to give the animals prior to cryopreservation that maybe, you know, would increase the efficacy and the quality, then you know, that would change it a little bit, right? Because we'd have to make sure that gets approved and so on, for humans as well, of course. But as far as, you know, the everything we do is after, you know, the point of clinical death, right? So so yeah, it helps because yeah, we don't get stuck sort of moving through all of that. Anytime you have to deal with the government, we're talking about very long timelines.
I understand. Yes, I absolutely understand. Well, you know, and there is a difference. You're kind of in that place as a startup, still in fairly early stage. That I always like what Elon said is there's a difference between R&D and production, a tremendous difference. So you're at that point right now. You're early stage. You don't really know what the beta site's going to look like, but it's exciting and good thing you don't have regulatory considerations, because, as you know, our foundation, the Puppy Up Foundation, we fund comparative oncology translational studies for cancer. And as you know, those clinical trials can cost so much money, take so many years. And it's so frustrating as a pet parent because we want access to that medication as soon as there's some demonstrable therapeutic benefit. I think that's why, to me, I believe I'm a true believer in N of one, right? If your kid is dying of cancer, that's my N of one, right? That's all I care about. So thank goodness that you don't have those tremendous, very expensive and timely regulatory considerations. But my guess is, though, when you do want to do your first beta site, the pet hospital that you're going to partner with is going to want to make sure that, especially if there are veterinarians that are involved with this, they're going to want to make sure that there's the partnership, that there's, I'm not sure, how is that going to work? Do you have a partnership in mind, or is that something that you're still working through with your first beta site?
Yeah, so there's, you know, like I was saying a little bit before, I kind of want our animal hospital cryonics facility hybrid to be an example. It's you know, I want cryonics to be in hospitals eventually.
Right.
Animal hospitals are kind of the first step there, and so, you know, the first animal hospital is just going to be ours. It's something we're building from scratch, from the ground up, right? It's not an existing animal hospital that we're partnering with. And we're using this as an example. And then what we're going to do is we're going to train, you know, veterinarians across the country that are in these animal hospitals that we partner with, to be able to perform the initial part of the procedure. So that's sort of the, you know, the blood washout, replacing with cryoprotectant and so on. And then, you know, that's sort of what I was talking about as far as bringing this to pets, right? As long as we have one animal hospital in every state, right? At least we can access everyone. So we've
had on our podcast a couple of times, and Ginger and I are both very good friends with Phil Bergman. Dr. Bergman, he runs clinical trials for all of VCA hospitals. And that's for all clinical trials. I mean, for all drugs, all disease states, not just cancer. And he would be, Dr. Bergman would be somebody, when you're ready to talk to, what does a partnership for a beta site look like? Do we need to do some trials prior to that? If you're interested, when we conclude this podcast, if you're interested in reaching out, I will talk to Dr. Bergman and connect you to, if you're interested. He's a man of science. And
Sure.
he is truly a man of science, and he would talk to you at length, be completely honest and upfront with, and let you know how they would look at it from VCA standpoint, if that's a bit of benefit to you, and if you're ready for it, Kai. You may not be there yet.
Yeah, no, I would love that intro. That sounds fantastic.
Yeah, like I said, I'm interested to see how this unfolds, and I'm so happy to be able to help you in any way I can. So we're running a little bit late, and I told Ginger I'm going to be yapping for a long time on this one.
Yeah.
I do every episode. I'm so bad, but I still have so many. I still have some questions,
No worries.
and especially in terms of risk. And I want to kind of go over that. Is that what do you feel like, Kai, would be if you talk to somebody like Dr. Bergman? One of the questions that he's going to ask is, where do you see the failure points? Where are the failure points, and what are the risks?
Yeah. Yeah. So, in the past, the cryonics facilities that have started, and a lot of them were founded in 1960, 1970, that kind of area where people started trying this initially. They've always been nonprofits,
Yeah.
right? I think, the idea there was maybe they can be a little more stable, maybe they can be a little more aligned with the mission. There's arguments both ways here as far as what's the best, for stability, what's the best for progress, and so on. But a startup has not been tried. A startup in the sense that we're taking outside external funding. We're moving very, very quickly, right? I'm not doing this over 50 years, right? This is something we're getting moving as fast as humanly possible. So there's, inherently there's quite a bit of risk there. I mean, all of this is unexplored territory, right? Even just from, not just from the structure, but just in general, trying to do this from a for-profit approach, trying to do this from trying to move fast and break things without,
Right.
Breaking things, right? And
Right.
No,
before you move on there, I'd like to stop you, because I have one of those brains that I go crazy. I go down rabbit holes, but I like what you said there, and I want to stick on that. It's something that Elon talks a lot about that wasn't true. It wasn't our model for doing things back when I was young, involved with high tech and biotech, is that we know we're going to fail. So let's just go ahead and fail fast, fail quickly. Let's pay for it. Hopefully don't fail too big or catastrophically, because the faster our failure rate, the faster that we're going to figure out solutions to it, and therefore we can make it commercializable, right? We can commercialize it, right? It's just the right way to do things, isn't it? It's the proper way for high tech development, technological development, don't you think?
Yeah, no, I mean, absolutely. I think, yeah, I mean, I think it's very easy to look at a problem and say it's impossible for that to be commercial, and this is just too crazy. People aren't gonna, but I think there is always a way. And if you can, hit that nail on the head, then I think progress really, really follows, right? Speed, progress. I think we can really take a field that's mostly been stagnant since it started, right? And really get it moving. So.
Yeah, exactly. And you're going to probably need some, I would say you're probably going to need some early adopters, some strategic early adopters, and some strategic, as JVs or partnerships. And I think that's why maybe a conversation with Dr. Bergman would be good, because if it's too early stage for VCA, he knows pretty much everybody what's going on in the field of science. So walk us through. I have one question though about, do we have storage? So let's say when I come to you, have you worked out, is storage there done? Storage is done in Utah, correct?
That's right. Yeah.
And so you already have the storage facilities ready to go, or that's something that you're building out as well?
Yeah, something we're building out. So it's been a lot of we've tried sort of off the shelf cryogenic dewars as far as storing the pets long term, right? Which you can get with pets, because with humans we're very tall, and you have to get custom made ones, but we can get ones off the shelf. And these are even behind me here, if you can see.
I did.
Are a few of the we likely won't use these in production, but they're just for testing a few different things. Not only procedure, but we're also making what we call dewar inserts. So there will be multiple pets in each dewar, and with animals that the size of them varies quite a bit. So we have sort of these dewars that are split up into 4 or 5 parts of the dewar that get sort of slid in. and those can strap in, whether it's a cat, whether it's a dog. And then likely right down the middle is where we have smaller animals, perhaps rabbits, and then hamsters and so on. The ones that we do use in production are likely going to be bigger than these. These could fit, maybe 3 or 4 dogs and then some smaller animals in them. We do want to get bigger doers just because the liquid nitrogen efficiency is better over time. But so yeah, we're working with custom ones and getting the right size that we need. But
Good. Well, Ginger didn't catch you at the beginning of the show. I did. You asked how large Lily was. So I think you were sizing Lily up to see if she was a candidate. Ginger didn't catch you, but you were trying to see if we can't think about that with Lily. Lily's going to live forever. I'm kidding. I always try to bring a little bit of humor. We are Fuzzybutts and friends. It's a tough conversation, but I want to remind the audience, and we can't say this enough because I'm going to make sure we get the accurate information about what you're doing out there. Is that there's no other alternative. That these are in termination situations. There are no other alternatives. When we speak of failure is not really an equation because there was no other alternative. So failure is we're having a different conversation. I think that's important because the pets that are candidates for cryonics, for cryo at your operation, they're end of life. There are no other alternatives. They have no quality of life. And we just hope that maybe science can figure something out very, very soon. If not, we don't know. But as you know, science moves so quickly that
we would never cryopreserve a healthy pet.
Right. Well, that's important, though. I think that's so important, though, Kai, because there are people in Canada that are euthanizing very healthy human beings, and I find I have a real big problem with that. And I also have I don't know if I should say this, Ginger, we might lose some audience, but I also have a problem with people who die and have their pets euthanized and buried, interred with them. You shouldn't be killing anything. Everyone wants to live in this world. We all want to live. All species want to live, and we should ensure that and do our best as possible, as best we can. We're stewards of this great big world, and so I have a problem with that. But that's why it's an important distinction, Kai, that everyone's listening should know, is that we're not talking about viable candidates. There's no other option. Death is the only certain option. Can we pause that biologically and then hopefully revive them in an intact state so that you can continue that relationship? I have a couple more issues that I have that are interesting, but just interesting questions, and it's so far down the line, but I wanted to throw them out there because I think they're interesting. Is that what happens if that timeline, that horizon is 20 years, and you have you're either gone or you're dead, and so there were aftercare and custodianship issues. And then what if it's only a few years and you've adopted a new family and you're like, oh, there's I'm kidding, of course, but there are those considerations, right? There's some really interesting considerations that I find very fascinating about your business model, right? Because there is a chance that a pet parent that I could pass away, and that, let's say Indiana, we cryo protect and preserve Indiana, and then I pass away for unknown circumstances, and then all of a sudden he's in one of the doers, he's there preserved there. So what do we do in that case? Or is it just too far down the road? You haven't dealt with that?
Yeah, no, I mean, these are very important questions. And it's almost like every question leads to another question when you're at a long enough timeline, right?
Absolutely.
I mean, what if, I mean, over 100 years or 50 years, it's so hard to predict the future.
Yeah.
I think, a lot of, what I talked about before as far as having cryonics in human hospitals eventually and things like that. I mean, I don't like death, and that's for pets. It's also for humans. In the past, people have been kind of, cryonics is kind of fringe for a reason. A lot of people see it as selfish, or there's just these kind of weird sort of views on it. And it's, you really can't blame people because the just communication in the field is just sucked for so long. And, but when it comes to pets, people are so much more excited, about it, and they're willing to do a lot of times they're not to do for themselves, right? For their pets. And I think, I really see this as almost a gateway drug for human cryopreservation. I want people to realize that Death is bad. It's a problem. It's something that we should be trying to solve. I think it's something that we can solve. And that's not just for our pets, that's for us as well, right? So over long enough timelines, you know, if you die in some sudden death manner and your pet's cryopreserved, I hope you guys are cryopreserved together, right? I hope that would be my you know, my long-term goal, and then I hope that you can be brought back together. But yeah, I think you know, and as far as, you know, short periods of time, right, if there's you know, some big sort of breakthroughs that allow us to bring these pets back sooner rather than later, there's, you know, there's considerations to take there, right? As far as, you know, most of these pets are dying of age-related diseases, right? So until we can reverse the aging process, it's likely not a good idea to bring these pets back from cryopreservation, because you'd be bringing them back in an old body, right?
Right.
So there's a certain level of, you know, technological advancement that we would want to get to before we bring these animals back, even if we can perfectly bring them back from cryopreservation without any issue. So just, yeah, there's a lot to, you know, consider there, but yeah, I think really the core thing at the end of the day that I want to get across is that, you know, we should be looking at death as a problem.
Right.
And for most of, you know, our time on this planet, you know, from being cavemen with sticks all the way up to now, it's been inevitable, right? But we're getting to the point where that might not be the case anymore. So, you know.
Yes, and there's so many, and there are, you're right, it's still a relatively obscure science, but there are so many very talented people working on this in tandem in different ways. And I had to make the interesting observation, you are a Peter Thiel-backed company, correct?
That's
I
right.
make the observation he's a backer of Elon. So I'm guessing Peter's hedging his bets. He's got Elon over here working on Neuralink. And so Neuralink, you can capture all the software and then put it into other hardware over here. And then he's got your company. Well, Peter seems to be an awfully astute investor and certainly an innovator in his own way.
You know, I mentioned persiflation earlier.
Yeah.
Peter was the one that funded that through Breakout Labs, right? So he's been interested in this field for you know, for quite some time, and I believe he's an Alcor member. I think he signed up for cryopreservation himself, and you know what, you know, I think he's definitely someone, yeah, who likes to hedge his bets and right, and sort of have his hand in a lot of different fields, but yeah, I mean, you know,
well, for other reasons though, because sometimes you don't know whether they're going to converge too, right? And that's another reason why to have invest in several companies approaching one problem or a similar problem from several different directions, is they might converge, and there's a synergistic opportunity as well, which I'm sure you know and Peter knows.
A lot of living a very long time may include having different, you know, transplants, you know, you got to get a new heart here, you got to get a new, you know, liver here, and so on. And, you know, in order to unlock that future, we need to be able to put hearts on the shelf and have them there for an indefinite amount of time, right? So even just organ cryopreservation, you know, really dips into some of the more, you know, more into the aging and longevity field. There's a lot of overlaps in these fields. So yeah.
Yeah, well, we got to make sure we cryo preserve Ginger's brain, because scientists need to study that for all of eternity. For sure. We've got lots to learn there. Oh my God, did I go there, Ginger? Is it nearing that time of the show where we start having fun? I'm kidding. I'm kidding.
Well, let me just tell you, talking about this, my feet are freezing.
It's getting pretty cold in the lab here too. Yeah.
Well, it's so exciting. You know, Kai, everything that you're working on, and the future is like envisions. I have an inkling of the world that you envision about these satellites, where you go into a hospital for whatever situation, and there's a crisis situation, and you can't come out, but thank God you can go and be paused. Right.
Yeah, there's
I love that scenario.
It's interesting because, you know, we will be, once we have the clinic up and the you know, cryonics facility hybrid, we will really, you know, it's almost like we'll be the first hospital in the entire world where if you need to get your dog's teeth cleaned and put under anesthesia, if you need to get a risky surgery for your dog, if you need to get a cancer treatment, you know, we'll be the only dog where you're maybe not risking permanent death undergoing those procedures.
Correct.
That may be a big inflection point for this work, as in, you know, would you rather go to the hospital that has this technology just in case, for pretty much any procedure that has some deal of risk with it, or would you rather go to the hospital that doesn't? And I think that's just going to be interesting.
That's a sell point for me. I just thought about that for the first time. I was thinking about other considerations, but it's absolutely a sell point. If a crisis situation occurs in this hospital, in that there is this other alternative that other hospitals don't, I would choose the one where, well, if nothing can be done. However, though, this is a sidebar. I got to make sure, damn sure that hospital is going to do everything that they can do, of course, before. But yeah, that's a tremendous sell point. I think I have an inkling of the vision that you and Peter see of the future for companion animals, and it's exciting. It excites me. And as we wind this down, Kai, I want to give you an opportunity. Is there anything else that you want to get in here and share with our audience that I may have missed about the wonderful technology you're working on?
I think we covered a lot. I think it's probably just worth mentioning that we do have the wait list open, right? So you can just easily.
Well, what's the website for that?
Yeah, it's just cryopets.com.
Okay.
And yeah, that's where you can find all of the science on there. We have blog posts, we have all kinds of things on there, so you can explore sort of more in depth what the procedure looks like, from start to finish and so on. But yeah, I would there's no you're not forced into it, right? It's just good to get your pet's names on it, and just in case for when that day might come. So yeah, that's probably all I would.
And if they sign up at cryopets.com for the wait list, do you send them updates about where you're at in the technology, what you're learning from all the R&D that you're doing, and keep everybody abreast of everything?
Exactly. Yeah.
Excellent. I'm going to do that as well. I'm very excited about it. And so you're also, I know you're Cryopets on Twitter or X now. Are you on any of the other social media nightmare platforms?
Yeah. Yeah, I would definitely recommend X, especially my personal account, which is just at Micah Mills. I exclusively post about the work that we're doing and sort of the way we think about things and all that. So
I follow it, and I love that you're transparent and you put the work up there as well. That's the future. All science should be up there, transparent. That's how you collaborate, so on and so forth. Unfortunately, dollars and cents have always gotten in the way of everything, scientific development since our existence. So, okay, great, Ginger. We've reached that time of the show as always. What else did I screw up? I know I've screwed up a lot.
I can't think of anything. It was perfect. Perfect.
You know why we do this. That's always your answer. I know I screwed some things up. There's some things up. So, but do you have a cancer tip for us this week? I know you try to do that every week for us. We try to help you out. Foundation
have a cancer tip. I'm always talking about having people check their dogs for lumps and bumps. And I'm not sure that I ever add in there, because one of the places people don't typically look is actually in their dog's mouth. And obviously dogs can get oral cancer. The most I have a little cheat sheet here because it's not something that I know a whole lot about, but typically the tumors are found in the roof of the dog's mouth or around the gums, which is something that I'm more familiar with. It grows quickly. And just some of the symptoms, I'm just going to go through some things to look for. If you haven't checked your dogs for lumps and bumps in their mouth, just if they start exhibiting any of these symptoms, you need to check and probably take them to the vet. So excessive drooling. I don't know, most peers drool a lot. Luckily, the ones in my house don't. Bad breath, difficulty chewing, blood coming from the mouth. If you think that they're having any oral pain, weight loss, loose teeth, a visible mass in the mouth, and large lymph nodes around the face and neck, or any swollen areas on their face. It could be a sign of something is wrong in their mouth, and you should have it checked out by your veterinarian.
Excellent. All good information. And yes, oral cancers, some of them are very aggressive and they're very deadly. And by the time you see a lesion in the mouth, it's already, in so many cases, metastasized. And unfortunately, that in so many cases, that's almost to the point where it's time to see someone like Kai, because while the outcome on certainly oral melanoma, as you know, Ginger, is so aggressive and advanced that the outcomes are very.
And that is the most common type of oral
Please
cancer.
check your mates' mouths and brush them. I'm not really good at that as a pet parent, but I do take them in to get dentals, and that's another way. But check the mouths. All right. So on that note, I would like to thank our very fascinating guest this episode, Micah Mills, with Cryopets. Thank you for being on Fuzzybutts and Friends.
Thank you so much for having me. Appreciate it.
You are welcome. And before you go, I wanted to give you an open invitation, Kai. You're welcome anytime back here to talk about progress in your research, to talk about partnerships, anything that takes you further and gets us to the next step. You're welcome back here anytime, my friend.
Amazing. Yeah, I'd love to come back maybe after we do the first case and, touch base, see where things are at.
Absolutely.
It
could be a shorter interview, just 15-minute update, however you want to do. The platform is yours because I'm a fan, and I know we're so early in this in early stage R&D, guys. We're going to get it out there and fail fast and fail quickly, and hopefully this is a viable solution for all our pets of the future. All right, everybody, I think we've gone over it enough. It was a lot of data for you, but I love this episode. It has so much promise for the future. Until next time, you can check us out on our iHeartRadio and our Spotify platforms. And you can also watch this live, which I recommend you do because Kai is sporting a Stevie Ray Vaughan look, and he's sporting it very well. I grew up in Austin, so I'm a big Stevie Ray fan. I'm not sure if that's the inspiration by the getup, but he's a great. You also get to see Cryopets, their facilities as well. So I do recommend that you check this out on our YouTube channel. It's fuzzybutstudios.com. Until next time, everyone, puppy up. Talk soon.
Bye.
Bye.